Good morning. Therapeutic Hypothermia, an ancient intervention with infinite potential by Justin and Kylee. I'm Kylee. This is Justin.
And I'm Justin. So therapeutic hypothermia. Today we're going to talk about some of the past applications, where therapeutic hypothermia originates from, kind of the history behind it. We're going to talk about some of the present protocols, the departments that are currently using therapeutic hypothermia, and then we're going to go into the future and kind of list off some of the cool things that are the studies and the research that's backing the future of therapeutic hypothermia.
Right. So a little bit about the history. In ancient Egypt, people wrote sort of some of the first medical protocols on papyrus paper that referenced using cool compresses and ice baths to treat painful swollen joints and inflammation. Fast forward just a couple hundred years to Hippocrates, a philosopher who was keeping journals and noted the difference between similar injury patterns in soldiers that were injured in the winter months versus soldiers who were injured in the summer months, and kind of compared those that the winter month soldiers tended to fare better and survived more often than the soldier months, and postulated perhaps that it had to do with temperature. In the Renaissance, the 18th and 19th century, a woman named Anne Green was hung and hung for 30 minutes, pronounced dead, and transported to the morgue. And by the time she got there, she had a pulse and was breathing, which prompted a gentleman named John Hunter, known as the father of modern surgery, to continue research in cold and hypothermic patients and resuscitation, which led to the development of ventilating the lungs and using electric shock therapy to restart the heart. And in the 1900s, lots of progress was made, starting with Dr. Temple Fay in 1930s. Dr. Temple Fay conducted research on cell metabolism and the effects of hypothermia on metabolism. And a gentleman named Dr. Safar did some research in 1958 on hypothermia in surgery and in cardiac arrest patients. All together, leading us to today.
So in 1958, this was a huge, instrumental year for therapeutic hypothermia. All of the research and the studies, from Hippocrates to Dr. Temple Fay to the Egyptians, everything was kind of leading up to this moment where in 1958 was actually the first human study of therapeutic hypothermia. So this was when they really got into what is happening in humans when we start to cool the body temperature. And they found out that 50% there's a 50% higher survivability rate in cardiac arrest patients when they're managed at a 33 degrees Celsius temperature. So that's actually a mild therapeutic hypothermia, and that's in comparison to a 14% survivability rate in a normal thermic patient. So you can see the huge jump there, right? That's what is that, like 36%? Like that's significant. And so this research really in 1958 is really what kind of pushed us towards looking into therapeutic hypothermia in a more in-depth way.
Yeah.
So we talked a little bit about the history, kind of where it originates from. Let's look a little bit into now where we're going to see therapeutic hypothermia implemented, what departments are currently using it, and you might not realize, but it's a lot closer and a lot locally. It's closer than you know.
It's more utilized than we realize.
Absolutely.
So thinking about presently, if we think about what hypothermia does, hypothermia essentially slows down the cell's metabolism. Well, a lot of what we learned from Dr. Temple Fay was hypothermic tissues require less oxygen, therefore requiring less ATP. So they don't have to go through the anaerobic respiration that a normal thermic high ischemic tissue would. to produce all that ATP. And that ultimately results in less oxidation and less cerebral edema, and cell death systemically. So currently, pre-hospital and hospital-wise, therapeutic hypothermia, or what they call targeted temperature management, is being utilized in cardiac arrest patients. But thinking about the pathology of cardiac arrest, what is that? It pretty much is just stopping the perfusion of tissue with blood and oxygen and full body ischemia. That led to Justin and I asking the question, what is the difference between cardiac arrest and a stroke, in the sense of cardiac arrest being systemic and a stroke being very specific. And right now, all we can do for strokes are recognize it fast, as in see the facial droop, see the arm drift, see the slurred speech, and recognize what time it started. And then recognizing CSAT positives for potentially large vessel occlusions, using neuroassessments to determine what cranial nerves are affected and what specifically in the brain is being affected by this ischemia, and then just transporting code 3 to the hospital, to the nearest appropriate facility, where they then can utilize fibrinolytic therapies and cath lab interventions by actually going in and removing clots and reperfusing that tissue. And all this time that it takes to reperfuse that tissue being normothermic, is producing all the lactic acid and producing all the oxidative free radicals that ultimately lead to the death of that portion of the brain and the not so good neurological outcomes as a result of stroke and the disabilities. So our thoughts were, is this a potential? Can we apply therapeutic hypothermia to stroke patients? And as of right now, there's a lot of obstacles.
Yeah. And so some of the equipment that we might see or be a little bit more familiar with in EMS and in the hospital, there's a lot of stuff out there that's being used and a lot of equipment that's currently being developed. Some of the stuff that you might be familiar with or heard of with therapeutic hypothermia in the pre-hospital setting, in the EMS setting, is as simple as like chilled crystalloids or chilled normal saline, right? The problem with that is by the time you get your drip set up and you start flowing those chilled fluids, by the time it goes through the drip set and it's now going through it's not a chilled drip set, it's now going through a room temperature tubing, it starts to warm up, and so you lose some of that some of that hypothermic or that chilled crystalloid by the time it gets to the vascular system. Another one that everybody knows about is just the simple external cooling with ice packs and getting the ice packs in the junctions, behind the legs, in between the thighs. But that's only as good as the longevity of your ice packs, right? Once your ice packs start to melt, well, now you're kind of, you're playing catch up now. And the problem with that is now the body starts to rewarm itself, and that's where the dangers lie. And a lot of the pathologies lie with therapeutic hypothermia is in the rewarming process. So what's awesome is actually Riverbend right here locally is using a couple different tools for therapeutic hypothermia. So they're using an endovascular cooling system, which is called the Zoll Intravascular Cooling Thermoguard. So what this is actually a catheter that goes in the patient, and it gives you a real-time live temperature reading of your patient, a core temperature reading of your patient, which is much more accurate. And it's going to adjust to the therapeutic hypothermia range, that the targeted range that you're trying to accomplish, whether that be a mild, moderate, or severe. Most of the time we're going to see the mild to moderate range. So this will this will counteract the rewarming and keep your patient at a steady therapeutic hypothermia range. Another one that they're using for external cooling is the Arctic Sun hydrogel circulating pads. So very similar to how a radiator works on a car and it circulates coolant. It's kind of the same concept. So these are pads that go on the patient, and it actually just circulates this coolant that holds a cold temperature much better than like water or whatnot, and it circulates around the patient. So super awesome. And then another thing that you guys might see sooner than later in the EMS setting is esophageal thermometer probes. Actually, in Vancouver, they are currently implementing this in their current protocols. And so when they place an ET tube, they also place an orogastric tube, and then they will go ahead and place a esophageal thermometer probe. And this is going to give them a much more accurate core temperature, and be able to titrate and adjust as needed, and know exactly, you know, where their patient's at as far as body temperature. So super awesome, already being implemented in protocols. So look out for that here in the future.
That and esophageal thermometer probes significantly easier to swallow than other types of probes. I had to say it. Y'all know what I'm talking about. So where do we go from here, right? So looking at all the obstacles and all the things in our way, we obviously don't intubate every stroke patient. So using esophageal thermometer probes, probably not going to work. And Justin touched a little bit on it, the rewarming process. Our body has natural defense mechanisms that are constantly fighting being cold. So some genius scientist or doctor or somebody asked the question, why is it that we're fighting being cold, and how can we better treat the shiver response and the cold stress response that we see in these patients that we're trying to cool for the neuroprotective effects? And that led to ultimately finding information on a potential solution that the literature calls hypothermia in a syringe, also known as pharmacologically induced hypothermia. Very, very cool stuff. Essentially, like I was saying, cold stress hormones, cold stress proteins, our body's natural defense mechanism against being cold, and then initiating the shivering and all the bad side effects, pulmonary edema, cerebral edema that we see in the rewarming process, and then how we could potentially counteract it. Currently being researched are neurotensin receptor 1 agonists. These act directly on the hypothalamus, also known as our temperature regulatory center in our brain. And then there's another study that's being done on thermoreceptors in our peripheral tissues that actually identify when you're cold and when you're warm, and then initiate the shivering and the cold stress responses there. I read a really interesting article talking about mice in TBIs and strokes. The scientists took these mice and essentially gave them strokes, and then injected them subcutaneously and intravenously, and compared the two modalities of application of the medication with a chili pepper derivative. And they found that when using the pharmacological induced hypothermia, you got a more stable and reversible hypothermic response. It was significantly less abrupt than the traditional cooling methods. It was more effective, and it kind of self-titrated, being subcutaneous, slowly being released into the body, slowly affecting the targeted receptors that it was trying to affect, and ultimately protected brain cells when they took scans of before and after in control and experimental groups. So very promising stuff, especially if it's just a subcutaneous injection, as simple as that. We do that on a pretty regular basis. So
absolutely. So as you can see here with the future, there's a lot of things that can be implemented into place, right? So moving towards this pharmacological therapeutic hypothermia in a syringe, and this, you know, is huge. That could be a game changer, because equipment is so big nowadays when it comes to therapeutic hypothermia, that if we can condense it, it makes it much more realistic to carry on an ambulance. But with that comes, you know, a lot of FDA approval, a lot of different hoops that we're gonna have to jump through to get this approved. So
right, all the clinical trials, all the FDA approvals. Yeah. First, we got to figure out therapeutic dosing, and is it safe? What are the side effects? That sort of thing. But ultimately, could be the answer to not only treating TBIs and strokes, and even potentially cardiac arrest patients and inducing hypothermia in those patients, but also in long duration space flight and putting people in suspended animation to fly them off to, say, Mars or other galaxies.
Oops. Yeah.
So in conclusion,
Yeah.
ultimately how and why,
Sorry, we had a minor
technological difficulties.
Yep. Don't you love technology?
No, I don't.
So yeah, and like Kylee was saying, in conclusion, what we talked about today is we talked about why and how therapeutic hypothermia works in the body, the physiology behind it, why it is so important. We talked about the history today, where it came from, where it was first implemented, and then we worked into the present. Where are we seeing it? There's current departments that are very progressive with therapeutic hypothermia, and then there's other departments that are not. And then like in the hospital, like initiating therapeutic hypothermia in the pre-hospital setting is beneficial if the hospital is going to go that treatment route. And then the future, right? Like Kylee was jacked up about this, the pharmacological side of things and therapeutic hypothermia in a syringe, like she said, an IM shot, like we do that all the time, and that's a game changer. But ultimately, the question is therapeutic hypothermia beneficial for these TBI stroke patients, for cerebral hemorrhage? We don't know yet. There's not enough research that's been done, but
yeah,
from what we have read, it looks very promising.
Right. We do know the science is there, that it protects the brain. It protects the brain cells, given the physiology of how it works, decreasing oxygen demand. And when those ischemic cells don't even need oxygen because they're cold, it results in a lot less cell death and a lot less disability. We do know that it works. As far as how to apply it, I think that's the biggest obstacle. And why I'm so jacked up about hypothermia in a syringe is that's a really easy application. It takes away the bulky, expensive equipment and the things that aren't realistic to put on an ambulance. And it gives us something that we've been pretty much giving this entire time, which is just a medication in a syringe. Yep. So it's interesting, has potential.
Yeah. Thank you guys so much for tuning in. I hope you guys learned something.
Looking forward to answering all your questions. Yes. And probably not having any of the answers.
Okay.
Right.
I gotta stop recording.